Good afternoon, sir. Good afternoon, everyone. Hello, sir. Am I audible to you?
Yes, ma'am. Yes, ma'am.
Good afternoon, sir. On behalf of Department of Microbiology at Yashwantrao Chauhan Institute of Science, Satara, I would like to extend a very warm welcome to you on this occasion of Faculty Development Program organized for the academic year 2021-22. So once again, I welcome you for this faculty development program. And now I request Dr. S.S. Kanse Madam to please introduce our resource person for today, Dr. Vishwas Patil, sir. So over to you, madam.
आधीच पीपीटी ओपन करून ठेवा खाली इनलाईन. हां. Hello. Good afternoon, sir.
Good afternoon, ma'am.
Okay. A very warm welcome on behalf of the Institute and the Department of Microbiology to the FTP program held today. So here I take the opportunity to introduce today's speaker, Dr. Vishwas Chormari Patil. He is an assistant professor and head department of microbiology, LBS College of Arts, Science and Commerce, Satara. He has been awarded with his PhD degree just recently in 2018. He has qualified GATE All India rank 501. He is net qualified with All India rank 41. He has cracked SET 3 times. He has done his MPhil also. He has been holding very responsible positions at university as well as at the institutional level. Has completed one minor research project of 1,70,000 granted by UGC Delhi. He has completed various courses of academic staff college, orientation and refresher courses, as well as completed many certificate courses. He has a vast research experience at MPhil as well as PhD level and published approximately 29 research papers and has out of 29, only one is in the National Journal. He has presented his research papers in various national and international conferences. And he has participated in various national, international conferences for paper presentation or poster presentation, as well as he has been a member of the organizing committee of various national and international conferences. So today we have with us a very young, enthusiastic personality, and I can say a well-qualified, well-studied personality who has a keen interest in the subject. I end my introduction of the speaker over here and I hand over the further responsibility to the speaker to start the session. Yes, Chormare sir, you can continue. Thank you
Thank you.
for accepting the invitation.
Thank you, madam. Thank you, madam, for a brief introduction. First of all, I thanks organizing department and Honorable Principal Sir for giving me this opportunity. On this FDP program, I will share my views and I will not guide you in the sense because we all are teachers and we are teaching this subject from many years and many of us have huge teaching experience. So I will begin with my presentation. Madam, will you please me help in sharing the PPT screen
Yes,
because I
yes,
have
sir.
used
Just a
Zoom
moment.
app. Okay, I have used Zoom app. I don't have experience with this.
Yes, sir. Just a moment. Sir, please try sharing your screen once again.
Uh, Where PPT is open. Okay, wait. Where is the option?
Sir, please share your screen by clicking the arrow at the side of right hand. Present now, this option will be there on your screen.
Yes, present now is there, but there are option your entire screen, a window, a tab.
Sir, you can directly start your entire screen as well.
But PPT is not visible there.
Sir, when you click on entire screen, the PPT which you have minimized will be visible at the,
Okay, it's going. It's working.
yeah, yeah. You can start with the PPT. You can just click on the PPT and it will be displayed on the screen. Yes, now it is visible, sir.
Okay. Okay, let's begin with the presentation. First of all, I really appreciate the efforts taken over by BOS team to design this curriculum for BSc first year microbiology subject. They have took into consideration the opinions of experts from academics, industry, and research fields. So this is a preamble for BSc 1 microbiology syllabus. These are the objectives of BSc program. So in semester 1, there are two theory papers, BMIT, that is BSc Microbiology theory paper number 1, that is 101 as a introduction to microbiology and scope, and second paper is BMIT 102, that is bacteriology, and this paper includes 4 units. Unit 1 is the bacterial cell organization, unit 2 is the microscopy, unit 3 staining and staining procedures, unit 4 is the control of microorganisms. And based on uh, the syllabus mentioned for these 4 units, these are the learning objectives which will be which will, the students shall be able to attempt. We'll see the syllabus and its learning objectives unit wise. So unit 1 in bacteriology paper is the bacterial cell organization, where we have to taught the morphology, size, shape, arrangement of bacteria, cytology of bacteria, which includes structure, chemical composition, and functions of cell wall, cell membrane, mesosomes, capsule, slime layer, and surface appendages as flagella and pili. So learning outcomes based on this unit 1 content is the after completion of the unit, the students will be able to study the size, shape, and arrangement of bacteria and study the aspects of gram-positive and gram-negative bacteria. So first subunit in unit 1 is the morphology of bacteria, where we have to discuss the size, shape, arrangement of bacteria. So this is the introductory part where bacterial shape and arrangement vary from species to species, and the study of size, shape, and arrangement of bacteria is helpful in classification and identification of bacteria. So first aspect is the size of bacteria, where the bacterial size ranges from 0.25 to 1 micrometer. The cocci-shaped bacteria have average range of 0.5 to 2 micrometer in diameter, whereas the rod-shaped bacteria have 0.25 to 1 micrometer range. Some bacteria possess diameter of 80 micrometer and length of 600 micrometer. The best example is Epilocysicum fuselsoni, which is a gram-positive bacteria which occurs in symbiotic association with surgeon fish. Among the bacteria, the smallest bacteria is Mycoplasma, having 0.25 micrometer diameter. The largest bacteria known so far is the Thiomargarita namibiensis, which is a gram-negative coccoid marine bacterium observed in Namibia region, which measures 0.1 to 0.75 nanometer size. So the basically size of bacteria is measured in terms of micrometer, and they are smaller than other eukaryotic cells like fungi. Shape of bacteria. The rigid cell wall decides the shape of bacteria. The common shapes of bacteria are cocci, bacilli, and spirillum-shaped bacterium. So these are three common shapes. And here you have to explain the characteristics of these three forms, and you have to also mention the examples while teaching. So here this slide presents the general properties of cocci, bacilli, spiral-shaped bacteria, and their respective examples. Then besides these three common shapes, other forms of bacteria are also observed in the nature. These include pleomorphic bacteria. The best example is the Rhizobium species. Others include Mycoplasma pneumoniae, Deinococcus rhabdoideans, Mycoplasma genitalum. These are pleomorphic bacteria. They exist in variable shapes. Then filamentous bacteria. The examples include Candidatus, Savagella, Nocardia, Amory, Alcalichins, Pimba, Thyatric species. Then there are star-shaped bacteria. The best example is Stella. Then there are box-shaped bacteria, appendaged bacteria, sheathed bacteria. So there are other forms of bacteria that are also observed along with the common shapes. Now see the arrangement of these bacteria. It is based on the type of cell division, and based on the type of cell division and cell arrangement and cell number, we'll study the arrangement of different kinds of microorganisms. First one is the arrangement of cocci. Here the arrangement of cocci is more complicated than the bacillus. And after the cell division, how they arrange with each other, based on this aspect, there are different kinds of arrangement. Those bacteria that exist as a individual, that is as a singular form, they're called as a coccus. Then diplococci. In diplococci, the cell division occurs in one plane, and two daughter cells formed. They remain in pairs. And the examples are Neisseria gonorrhoeae, Diplococcus pneumoniae, etc. Then in streptococci also, the cell division occurs in one plane, and the newly formed cells, they remain attached to each other in a single direction only, and they form a chain of streptococcus. The examples are Streptococcus pyogenes, Streptococcus mutans, Streptococcus mitis. In tetracoccus arrangement, after the cell division, which occurs in two planes, the newly formed cells remain joined to each other, and they form tetracoccus arrangement. And the examples is Gafkia tetragena. Then in Sarcine arrangement, the cells appear as a group after the cell division, which occurs in three planes, and after the division they remain. This division occurs in regular pattern. So the examples are Sarcina lutea, Sarcina ventriculus, and Sarcina aurantica. Then in Staphylococcus, which is a grape-like arrangement, it is formed due to the cell division that occurs in three planes but in irregular pattern. and the cells that are formed, they remain attached to each other, and they form these cluster-like appearance. So these are the different kinds of arrangement of cocci and their respective examples. Then arrangement of bacilli. On the basis of arrangement of bacilli, these bacilli, there are of these types: singular bacilli, then cocobacilli, where they are observed as a oval and rod-shaped bacteria. Then there is a diplobacillus, where cell division occurs in one plane and newly formed cells remain attached to each other, diplobacillus. Then streptobacillus, there is attached arrangement of newly formed rod-shaped bacteria in a chain. Then palisade-like arrangement, it is the arrangement in a side-by-side sequence, like matchsticks appearance. The example is Corynebacterium diphtheria. Then trichome. The trichome-like arrangement is similar to the streptobacillus, but here in trichomes, the surface contact that is observed in between adjacent cells is high. Then arrangement of spiral-shaped bacteria, they usually exist in single, and on the basis of
Sir,
arrangement
sorry
and
to interrupt you.
okay.
Sir, please will you click on the hide option so that we can see the entire content of the slide? Yes.
Okay,
Thank you,
sorry,
sir.
ma'am. Okay, sorry.
Yeah. Yeah.
So arrangement of spiral, it is in the three forms: singular spiral shaped bacteria, then spirochetes, then vibrio. In vibrio, so these spiral shaped bacteria, they have rigid spiral structure and they exist usually as a singular form. In the spiral shaped bacteria, twisting occurs within the cell, and on the basis of twisting, there are two types of bacteria. There is a vibrio, they appear as a comma shaped and form after one complete twist. And there are different species of, observed among the Vibrio genus: Vibrio cholera, Vibrio fulnificus, etc. Then spirochetes, it is formed after 2 to 3 complex twists. So these spirochetes, they are highly flexible in structure. The examples are Spirocheta species, Treponema species, Borrelia species. Then after the morphology, let's turn towards the cytology of bacteria cell, where you have to discuss with your students the structure, chemical composition, and functions of cell wall. So cell wall is the outermost component of a bacterial cell, and on the basis of differences in their cell structure and chemical composition, there are two types: gram-positive and gram-negative bacterial cell walls. So this table describes the differences in between the gram-positive bacterial cell wall and gram-negative bacterial cell wall. So you have to discuss these differences with the students. Then right hand side image is explains the structure of a bacterial cell wall. First is the gram-positive cell wall, where you observed the cell wall which is situated next to the outer side of a cytoplasmic membrane, and it is a single thick layer in case of gram-positive bacterial cell. And here, this single thick layer is composed of uh, peptidoglycan and teichoic acids. Teichoic acids, and this peptide chain, it is attached to the cytoplasmic membrane by uh, number of lipoproteins. So these are the three basic components, that is peptidoglycan, lipid teichoic acid, and lipoproteins that are observed in the gram-positive cell. Then in gram-negative cell wall, it is double-layered cell wall, inner layer and outer layer. Inner layer is present in between cell membrane and outer membrane of a cell wall, and this inner membrane is composed of peptidoglycan layer, and outer membrane is composed of a regular uh, it is similar to the composition of a cell membrane, except it contains the lipopolysaccharide molecules. So again, this slide represents the components of a bacterial cell wall, where cell wall of a gram-positive bacteria is composed of peptidoglycan, teichoic acids, and lipoproteins. And the cell wall of gram-negative bacteria, it is more complex than the cell wall of a gram-positive bacterial cell. So these are the components. Inner layer is composed of peptidoglycan, outer layer phospholipids, lipopolysaccharide, proteins, and lipopolysaccharides. So you have to discuss these components, their structure in detail, and their chemical composition and their functions. So these are the some images of bacterial cell wall, gram-positive bacterial cell wall, and gram-negative bacterial cell wall. So the composition of a cell wall, these are the components of a cell wall. Number one is the peptidoglycan. Here you have to mention the properties, the structure of peptidoglycan. Here it is a polymer of N-acetyl muramic acid and N-acetyl glucosamine that are linked together by beta-1,4 linkages. Then we see the tetrapeptide side chain which is attached to the N-acetyl muramic acid, that is L-alanine, D-glutamic acid, meso-DAP and D-alanine. So this a tetrapeptide side chain, it is attached to the N-acetyl muramic acid, and this is the tetrapeptide side chain responsible for the cross linkages in between the peptidoglycan of a uh, similar polymer or adjacent polymers found in the cell wall. So there are different types of cross linkages. Here in diagram C labeled, it is a peptide linkage that is observed in between DL9 and meso-DAP. So there are different types of linkages that occurs in between the structure of peptidoglycan. You have to also mention these types in detail. Then second component is the lipoticoic acid, or it is also called the lipoticoic acid, which are exclusively found in gram-positive bacteria, and they extend through the entire peptidoglycan layer, and they appear on the cell surface, and they contributes to the antigenicity of a bacterial cell. So there are two types. You have to mention the types, and you have to also describe the structural aspects and their properties as ribitol teichoic acid and glycerol teichoic acids. Then their functions are also to be discussed in detail. So this was the second structural component in gram-positive bacterial cell wall. Then third is the lipopolysaccharide, which is exclusively observed in the cell wall of a gram-negative bacterial cell. It is present in the outer membrane of a gram-negative bacterial cell wall. If you look at the structure of lipopolysaccharides, it possesses three components. The basal component is called as a lipid A, middle component is called as a core polysaccharides, and upper portion that is excreted from the cell wall is called as the chain of oligosaccharides, which is immunogenic in nature, and hence it is called as a O antigen. Then this lipid A is responsible for endotoxicity of a lipopolysaccharide molecule. So this molecule is exclusively found in gram-negative bacterial cell, and you have to mention, you have to describe the structure of lipopolysaccharides, and you have to discuss the functions associated with different components of these lipopolysaccharide molecules. Then phospholipids, they are present in the outer membrane of a gram-negative bacterial cell wall. The composition of phospholipids is similar to those found in cell membrane. They form a bilayer leaflet, and these phospholipids, they possess hydrophilic head and hydrophobic tail. So this polar head, it is situated at the top portion, and non-polar tail, uh, they aggregate to each other and they are stabilized by hydrophobic interactions, and hence these structures, whenever they are mixed together, they form a lipid bilayer structures. Then next component is the proteins. A variety of proteins are found in the cell wall. Most of these proteins differ from that found in the cell membrane. So you have to describe various types of proteins and their functions that are found in the cell wall. The proteins that are found in the cell wall, we call it as a porins because they are found as a they're the aggregates that forms channels, and these channels, they serve for specific transport and also for non-specific transport. So you have to explain the different functions of these porins molecules while teaching. Then lipoproteins molecules, that was called the Brown's lipoprotein. They are embedded in the inner side of outer membrane. They covalently links underlying peptidoglycan layer and outer membrane of a cell wall. Now the last part of a cell wall is the functions of cell wall. There are different functions of cell wall. Some of them are mentioned here. It gives shape to the cell. It gives protection against osmotic pressure. It is essential for cell growth and division. It allows entry of nutrients and exit of waste products. Then different components They're responsible for antigenicity of a cell wall. They're described here, oocyte chain, then teichoic acids. Then LPS molecules, they are endotoxins and serve as a virulence factor for gram-negative bacterial cell. So these functions that we discussed. Then second cell organelle is the cell membrane, where you have to discuss the chemical composition, structure, and functions of cell membrane. In chemical composition, the cell membrane is composed of membrane lipids and proteins. Here, the membrane lipids, they are again of two types, polar lipids and non-polar lipids. And the major portion of the membrane lipids are polar lipids. And they include sphingomyelins, glycerophospholipids, which also includes lecithin, cephalin, lipositol, phosphatidylserine. And non-polar lipids, they include cholesterol, triglycerides, and glycolipids. Then membrane proteins, they are extrinsic proteins that are present at the peripheral region of a cell membrane, and others are intrinsic proteins. There are major portions of membrane of proteins. In structure, there are different structures. There are different models that explain the structure of a cell membrane. One of them that is widely accepted is a fluid mosaic model proposed by Singer and Nicholson in 1970. And according to this model, you have to explain the structural components and their functions. The structural components and their functions. So this is a model. Uh, this is an image that explains the different structural components. You have to explain the properties. Then you have to explain the structure of each component and their functions. Then functions of cell membrane. It is a semi-permeable barrier of a cell. It serves as a principal osmotic barrier of the cell. It acts as a site of biosynthesis for cell constituents like capsule and cell wall components. It gives attachment site for chromosome and plasmid. Electron transport chain is located in cell membrane of aerobic bacteria. Photosynthetic apparatus in case of purple bacteria is present in the cell membrane. Then next cell organelle is the mesosome, which are nothing but enfoldings of cell membrane, which provides the site for attachment of chromosomes, and they also plays an important role in cell division. So you have to explain the structure of these mesosomes and functions in detail. Then we have bacterial capsule. You have to explain here the location of a capsule, then chemical composition of a capsule, then differences in between capsule and slime layer. So here in image, the capsule, we all know that it is located outside the cell wall. Then in chemical composition, the major portion of a capsule, that is 98% is represented by water, and 2% are organic components, which are polysaccharides and polypeptides. So there are some bacteria that possess capsule that is solely made up of proteins. The best example is Bacillus anthracis. And among the polysaccharides, there may be homopolysaccharides, that is polymer of only one type of sugar, glucan and levain. And there are also capsules that are produced by some organisms. They are composed of heteropolysaccharides. So you have to also mention here the examples of bacteria that possess the capsules constituted of homopolysaccharides and heteropolysaccharides in nature. Then differences in between capsule and slime layer. You have to explain these differences in detail. This table represents the differences in between capsule and slime layer. I think there is no need to go in detail because everybody we have learned these aspects while we were students and we were teachers. We were teaching for many years. Then functions of capsules. It gives temporary protection against drying because it contains 98% water. Then it is antiphagocytic, hence governs virulence factor to a cell. It helps bacteria for attachment of biofilm formation. It is reservoir for certain nutrients because it is composed of polysaccharides and proteins. Then it gives protection to cell wall from antibacterial agents like lysozyme and phages. It acts as a barrier. Then it's serve as a K antigen and VI antigen. Then bacterial capsules are species specific, and hence they can be used for immunological detection and distinction of closely related species, and hence it is used in diagnosis. Then surface appendages, flagella and pili, they are to be discussed. The flagella, it is a locomotory organ of a cell. These are the some general characteristics of flagella mentioned here with, where size is mentioned here, its diameter. It extends from cell membrane through cell wall. Then these flagella are so thin, and hence we stain these flagella by special staining procedures. There we increases their thickness and we observe it under light microscope. And the detailed structure of this flagella can be observed in electron microscopy. Then you have to explain the structure of flagella. So we all know that the flagella structure is composed of three components: a basal body, which is associated with the cytoplasmic membrane and cell wall of a cell, a basal body. Then there is a short hook and a helical filament, which is made up of flagellin protein. So these are the three portions. You have to discuss these three components in detail. Then these three arrangement of these three different parts of a flagellum to a bacterial cell via cell membrane and cell wall is mentioned here with. So you can come to know, or students can also get exact idea how they are attached in cell membrane and cell wall, and how they function. Then on the basis of flagellar arrangement, there are different types of flagellar arrangement found in bacteria, and on the basis of this flagellar arrangement, the bacteria are classified as a monotrichous flagellated bacterium, where a single flagella is observed. The example is Vibrio cholerae. Then locotrichous, where more than one flagella are observed at one end only. Pseudomonas fluorescens is the example. Then an amphitrichous flagella, where a single or more than two flagella at both the ends. The example is Aquaspirillum serpens. And peritrichous flagellated bacteria, where you will see the flagella all over the body. The example is Salmonella typhi, E. coli, Proteus vulgaris. Then functions of flagella, it is responsible for motility of the bacteria. We have to mention here the mechanism of motility. Then it serves as a receptor site for attachment of bacteriophages. Then it serves as an antigenic determinant of bacteria, where the flagellar antigen is called as a H antigen, that is Hosh antigen. Then next, surface appendages are pili and fimbriae. These are mostly similar. This here in this slide, the pili and fimbriae, their properties are mentioned and their functions are also mentioned. The differences in between these two, they are given on the next slide. Here, these are the images of pili and fimbriae, and the next table explains the differences or comparison in between pili and fimbriae. Where appearance, pili are hair-like straight appendages, whereas fimbriae are tiny bristle-like fibers arranging from the surface of a bacterial cell. If you look at the length, pili are longer than fimbriae, and these fimbriae are shorter than pili. The number of pili per cell are 1 to 10 per cell, whereas fimbriae are numerous, 200 to 400 per cell. Then presence, these pili, they are present only in gram-negative cell, where these fimbriae, they are found in gram-positive and gram-negative bacterial cell. And if you look at the composition, these pili are composed of pilin protein, and fimbriae, they are composed of fimbrillin type of proteins. So this is about the morphology and cytology of bacteria. So let's turn towards the second unit, that is microscopy. And in microscopy, we have to discuss the types of microscope, then types of light microscope, then the light microscope, where its principal construction, working, and applications, and also the similar points related to the electron microscope, then the comparative study of compound and electron microscope. And after the learning this unit, the students will able to learn the different types of microscope and working and principle of light and electron microscope. So second unit, that is microscopy. You have to explain here the introduction, that is the definition of microscope and importance of microscope. Here microscopy, we all know that it is a study of microscope, and microscope is nothing but an optical instrument consisting of a single lens or combination of lenses that are used to magnify the size of a minute objects. Then importance of microscope, as we know, the microorganisms, they cannot be visualized. Microorganisms are those organisms that cannot be visualized with naked eyes because the limit of resolution of human eye is 0.2 mm, that is 200 micrometer. So those objects that have size less than 0.2 mm cannot be visualized directly with the naked eyes, and hence to observe these objects we need the microscope. So let's see. Then you have to discuss the types of microscope, and there are two approaches. There is first approach is on the basis of number of lenses. We have two types, that is simple microscope and compound microscope. The simple microscope consists of a single lens, whereas compound microscope contains two lenses. Then second approach is on the basis of principle of magnification, source of illumination and principle of magnification. Microscopes are of two types, light microscope and electron microscope. We'll see, we'll discuss in detail in next few slides. The next approach is the types of light microscope, and here in our syllabus there are four light microscope. There is bright field microscope, where the object that is to be observed appears dark against the bright microscopic background. In dark field microscopy, we see the dark microscopic field, whereas the object that is in focus, it appears as a bright. In fluorescence microscopy, the fluorescent dyes are used to stain the objects, and the object under study, it appears fluorescent. And in phase contrast microscope, the objects that are to be resolved, they possess different refractive index, and hence phase contrast microscope differentiates these two components that have variations in their refractive index, and they are clearly visible here. Then in types of electron microscope, we have transmission electron microscope and scanning electron microscopes. Then you have to study the parts of a light microscope. We all know these are different parts of a microscope. This will be further discussed in next slide. Some useful concepts in microscopy. Number one basic concept is the image formation in compound microscope, where we all know that in compound microscope we use visible light. So visible light, it is reflected by a mirror towards the condenser. Then this condenser converts them into the parallel beam of light. Then iris diaphragm controls the intensity of light that is to be focused onto the object that is present on the glass slide. Then whatever the light rays that are focused onto the object on the glass slide, most of the light rays, they are reflected back, and those light rays that enters into the objective, we call them it as a refracted light rays, and they are passed from the objects. So these refracted light rays that are coming from the object, that are collected by this objective lens, and it is a convex lens. It collects these light rays, these refracted light rays, and it forms a primary image which is real and inverted. And based on the magnification power of this objective, it is accordingly magnified. If there is 10x, if there is 10x objective, then there is 10 times enlargement. If it is 45, we will see the 45 times magnification. So this is a primary image which is real and inverted, that is shown here, image, primary image. Then this primary image is further magnified. It is passed through body tube, and it is converted by eyepiece into a final image which is virtual and erect image. virtual and erect. So in image formation, there is a magnification at two levels, that is at objective and at eyepiece level, and final image that is observed, it depends on the magnification power of a objective and eyepiece. Then magnification, this is the next basic concept in microscopy. This magnification is nothing but a degree of enlargement, and this magnification depends upon the distance of the object from the focal point. Closer the object to the focal point, greater will be the magnification. But if you try to uh, place this object uh, at at nearly distance, then it produces a defects. It produces a defect. So the distance in between the object and focal point, that is focal length, it is a constant. So this is a first parameter, that is distance of the object from the focal point. Then second factor, that is the curvature of lenses. The smaller the curvature of a lens, greater will be the magnification. And in a compound microscope, as I said, this magnification, it is carried out at two levels. So compound microscope produces magnification at two stages: objective, and depending upon the magnification power, the objectives are low power, that is 10x, high power 45x, and wall immersion lens, that is 100x. Then eyepiece 10x. So the total magnification of microscope equals to magnification power of objective into magnifying power of eyepiece, and then 100 into 10, 1000x. But this magnification can be increased if you use 15x as a uh, 15x eyepiece. If you use the 15x eyepiece, then this magnification can be increased to 1500 times. Then numerical aperture, you have to define here the numerical aperture. Then the brightness of an image and resolving power of a microscope depends on this numerical aperture. The definition is, it is a product of refractive index of a medium between objective lens and the object, and angular aperture, half the angular aperture. So the formula for determining the numerical aperture is n sine theta. And here theta is the angular aperture. Angular aperture means the angle made by the light rays that are extreme, that are wider. So the angle made by wider range that come out of luminous object and enter into the objective lens is called as a angular aperture. So depending on this formula, you have to calculate the numerical aperture, and the numerical aperture for a numerical aperture, it comes 1, and this numerical aperture, it is responsible for brightness and a contrast, and this numerical aperture can be increased above 1 by placing a drop of cedar wood oil, where this numerical aperture is increased to 1.5. Actually here, for oil immersion lens, the numerical aperture that is calculated in it is 1.5, but it is 1.4 because sine 90, there is no exact 90-degree angle made in between this. So this numerical aperture, actual practical, it is 1.4. The next basic concept is the resolving power. So it is the ability to resolve two adjacent points as separate and distinct. So this is a definition of resolving power. Here the formula is resolving powers equals to lambda by 2 into numerical aperture. So you have to describe this formula, and according to formula you have to calculate the resolving power. And for light microscopy it is 0.21 micrometer resolving power. So while discussing with the students, this resolving power is to be discussed with respect to definition, formula, then calculation for resolving power for light microscopy. And here the limit of resolution of light microscopy is 0.2 micrometer. Then working distance is the distance in between the front. It is the distance between the front of the microscopic objective lens and the surface of the specimen or slide cover slip. Whenever the object that is under study, it is in sharp focus. So it is called the working distance. The table represents the working distance for low power, high power, and wall immersion lens objectives. So these are the basic concepts. Then you have principal construction, working, and applications of bright field microscopy, that is compound microscope. Here you have to explain the principle of light microscope, that is compound microscope. Then construction of light microscope, and we all know that the light microscope, that is compound microscope, the components are divided into three parts: mechanical parts, then magnifying parts. There are two lenses that plays an important role in magnification, objectives, and eyepiece. And here you have to discuss their uh, properties also, and there are different. You have to explain the different properties of these objectives and eyepiece. Then illuminating parts, they plays an important role in illumination, that is focusing the object, mirror, iris, diaphragm, and condenser. You have to explain their roles here. Then working of light or compound microscope. This is a ray diagram that explains the working of light microscopy. And here, as I said, ray diagram, it begins with the mirror that reflects the light towards the condenser. These parts, they are not shown in the figure. So in working, you have to explain the role of mirror, condenser, then iris, diaphragm, then object, then uh, the role of objective, then role of eyepiece, and the final image that is observed. It is a product of magnification at two stages. And here, during this compound microscope, the magnification is achieved at two levels, two stages, at objective and at eyepiece. And hence, the primary image that is formed at objective lens, you have to tell them it is a real and inverted, and in eyepiece, it is virtual and erect image. Then applications of light microscope. There are different applications. Some of them are mentioned here that we routinely teach to our students. Then electron microscope. Here, introduction. You have to explain the introductory part of electron microscope, and here the importance of electron microscope, because in light microscopy the limit of resolution is zero point 0.2 micrometer, that is 200 nanometers, and hence those objects that have size below 200 nanometer, their observation needs electron microscope. And here the range of objects that can be clearly visualized in electron microscope is zero point 0.5 nanometer to 200 nanometer. That is, the resolution limit of the electron microscope is zero point 0.5 nanometer. So this electron microscope works in a range zero point 0.5 nanometer to 200 nanometer sized objects. Then this electron microscope, there are two types: scanning electron microscope, SEM, and TEM. Then first, scanning electron microscope. Here is an introduction, and this is a scanning electron microscope, and ray diagram is mentioned there. Then principle of SEM. In principle, in scanning electron microscope, the source of illumination is the electron beam that is focused from electron gun. And this electron gun generates the electrons with very high speed and acceleration. And these electrons, they are passed onto the object that is to be scanned, that is to be studied. And here, the medium of traveling this source of illumination is the electromagnetic waves, because the electrons, they may deviate from their routes, and hence electromagnetic field is used here. So electron beam that is generated at high speed, it is focused onto the specimen with the help of condenser and condenser lenses, which are nothing but electromagnetic material. And then the surface that is incident with this beam of electrons, the electrons, they are emitted from the specimen, and this, depending upon the structure, topographical structure of a object, they are differentially scattered from the surface. And hence here these scattered electrons, they are captured by secondary electron detector, and then it forms a image. Construction and working of SEM, these are the components: electron gun, then the condenser lens, then scanning coils, then condenser aperture is there, then objective lens is there, which plays an important role in magnification, then scanning electron detector is there, then sample is there, and after once the electrons they are bombarded onto the sample, the reflected electrons they are captured by scanning electron detectors and they produce a image. Then you have to mention the advantages of SEM, disadvantages of SEM. Then next type of electron microscope is the transmission electron microscope, which plays an important role in determining the ultrastructure. Here, in principle, here the red marked portion is a it explains the actual mechanism of working of transmission electron microscope. The electron beam that passes through the specimen and electrons they are scattered depending upon the thickness or refractive index of different parts of the specimen. The denser region in the specimen scatters more electrons and therefore appears darker in the image since fewer electrons strike the area of the screen. In contrast, transparent regions they appear as a brighter. And here in transmission electron microscope, staining with staining the specimen with heavy metals increases the contrast. It increases the contrast. And these stains they may be either fixed on the specimen, we call it as a positive staining, or they can be deposited outside the object that is to be studied, that is we call it as a background staining. And this staining helps in more contrast. Then the electron beam that comes out of the specimen, they're, uh, they're collected by the objective lens and they form the image. And then the final image, it is made visible onto the phosphorescent screen. Among the components of TEM, the components are similar to SEM. There are additional lenses, that is projector lenses, which is equivalent to the eyepiece. But here, the final image that we get from projector lens, it is directly uh, focused onto the phosphorescent screen, and the final image is made visible. Here, in components, there is electron gun. Then there are electromagnetic lenses, condenser lenses is there, objective lens is there, projector lens is there. Then we have here specimen holder, which is made up of a copper metal grid, which is transparent to the electron passage. Then finally, we have image viewing window or recording system. Then applications of electron microscope. Several applications are there. Some of them are mentioned here: to observe the viruses, to measure the size of viruses, to study bacterial flagella, intracellular structure observation, localization of enzymes, determination of concentration of colloidal particles in suspension. Then last part in microscopy is the comparative study of compound and electron microscope. These are the ray diagrams for light microscope, transmission electron microscope, and scanning electron microscope. The actual mechanism of magnification is similar in all these three microscopes. There are differences with respect to these points or characteristics we will study here. In compound microscopy, the source of light is the visible light, whereas in electron microscopy, it is electron beam. The medium of transmission in light microscopy it is air. In electron microscopy, it is high vacuum. Nature of lenses: glass lenses are used in light microscope. Electromagnetic lenses are used in electron microscope. Focusing mechanism is lens position is adjusted mechanically in compound microscope, where there is current adjustment in electron microscope. Then changing the magnification involves switching the objective lens, whereas here in electron microscopy it involves adjusting the current to the magnetic lens. The source of contrast or actual the mechanism is differential light absorption by the object that is responsible for contrast and observation. Whereas in electron microscopy, scattering of electrons is the basis for contrast. Specimen is mounted on glass slide in light microscopy. It is on metal grid in electron microscopy. Best resolution is zero point 0.2 micrometer, whereas in electron microscopy it is 0.5 nanometer. Highest practical magnification 1000 to 1500 times magnification is achieved in light microscopy, where it is over 1 lakh magnification. Affordability, light microscopes are cheaper, where electron microscopes are expensive. So this was about microscopy. So you have to discuss these points with the students in detail. Then unit 3 is the strain and staining procedures, and these are the learning outcomes related to this third unit. We'll briefly go through these points: definition of stain and dye. So stain and dye, they have all their organic compound containing both chromophore and exochrome groups that are linked to the benzene ring. So stain and dye, they are chemically same, but they differ in their use, purity, and concentration. So basically, they contain chromophore group and oxochrome group. This chromophore, this chromophore group is responsible for coloring property of a stain, and oxochrome group is responsible for its electrolytic dissociation and attachment to the object. So here there are the differences. They are used to color of biological objects, whereas dyes are used for non-living materials. Stains are pure and carefully manufactured. Dyes are not as pure as stains. Then these stains, they are used in low concentration, usually below 1%, whereas dyes, they are used in high concentrations. These stains are expensive, whereas dyes, they are less expensive. Then you have to explain the structure of stain with the students, that it consists of benzene ring, chromophore group, and oxochrome group. So benzene ring with only chromophore group is called as a chromogen. So here, the first structure, trinitrobenzene, it is a chromogen, and it has a yellow color, but it do not have ability to fix onto either living or non-living materials. So it is a chromogen. Then picric acid, it is a dye because it has color and it also has ability to fix onto the object. And this is due to the electrolytic dissociation of this picric acid, where the chromophore group is nitrate and oxochrome group is OH group. This oxochrome group undergoes electrolytic dissociation, and it forms picrate ion, which has negatively charged. So it is a acidic stain or acidic dye, and it will attach to a object that have positive surface, positively charged surface. Then you have to explain the classification of stain. So based on the charge present on oxochrome group, these stains, they are classified accordingly. In acidic stain, the oxochrome group, after electrolytic dissociation, it has negative charged. So they are called as acidic stain. They are used to stain positively charged components of a microbial cell. And the examples of acidic stains are eosin, nigrosin, India ink, acid hoxin, and congarate. In basic stains, they have positively charged oxochrome group after their electrolytic dissociation. So it is also called as a cationic stain. In these basic stains, they are positively charged stains. They are used to stain negatively charged components of a microbial cell. The examples of basic stains includes methylene blue, safranin, malachite green, basic fuchsin, and crystal violet. Then neutral stain, it is complex salt of dye acid with dye base. That is, it possesses both the charges. It stains both positive and negative charged components of microbial cells. The examples of neutral stains are Giemsa stain, Limsa, Leishman stain. Then study of bacteria by staining. We all know the staining procedures. Initially, we prepare a smear on a clean grease-free slide. Then we dry it in air. Then we heat fix it. Heat fixation is done in order to kill the microbial cells, and these microbial cells, they are also firmly attached to the glass surface. Then after heat fixing, we subject it for staining by flooding the slide or smear with a suitable stain. Then we keep it for 1 to 3 minutes. Then we rinse it, dry it. We put a cedarwood oil drop over the smear, and we observe it under microscope. So study of bacteria is done by staining and microscopy. Because bacteria, they are colorless, they are minute sized objects, and hence staining increases the contrast in between the bacteria. The common staining techniques, simple staining and differential staining. This table explains the differences in between simple and differential staining. In simple staining, only one stain is used, whereas in differential staining, more than one stain are used. Simple staining, it imparts only one color to all bacterial cells, whereas in differential staining, two or more different colors are observed onto the bacterial cells. Then, simple staining reveals the size, shape, and arrangement of bacterial cells, whereas differential staining reveals size, shape, arrangement, and also differentiate in between two groups of bacteria. Simple staining includes monochrome staining, methyl blue staining, etc. Then differential staining includes gram staining and acid fast staining. So the first staining procedure is the simple staining. It is also called as a monochrome staining or direct staining or positive staining. We all know the meanings of these terminologies. So while teaching, you have to define each of these terminology, that is simple staining, direct staining, positive staining, monochrome staining. Then you have to explain the principle of simple staining. Then you have to explain the procedure, observation, mechanism, and applications. Then second staining is the negative staining. It is also called as an indirect staining or relief staining because in this staining technique we stain the background and the actual object or the bacteria that is to be visualized, it remains unstained, and hence it is called as a negative or indirect or relief staining. Then here you have to explain the procedure, then also explain the observation mechanism and applications. In procedure, we took a drop of nigrosin stain at one corner of a slide, then we mix a loopful of bacterial suspension into it. Then with the help of another slide, we prepare a sorry. With the help of another slide, we prepare a thin and uniform smear. Then we air dry this smear, and then we put a drop of cedarwood oil over a smear and we observe it under oil immersion lens. Now see the mechanism. The bacterial cell surface is negatively charged because of the presence of carboxylic groups there, and the in negative staining, this nigrosin is an acidic stain, and it is an acidic stain. And usually, we all know that acidic stains are used to color the positively charged cell components. But here, the cell wall is negatively charged, and hence this nigrosin will deposit over the cell wall, and hence the bacterial cell will remain colorless, and the background will appear colored. Then you have to explain their applications. So in staining procedures, you have to go by defining a particular staining method, then its different names if they exist, then you have to explain the procedure, observation, mechanism, and applications of these staining techniques. Then differential staining. Here again, you have to define the differential staining procedure. Then there are two types of differential staining, gram staining and acid fast staining. Here first is the gram staining. It was developed by Hans Christian Gram in 1884. Then it went different modifications, and the today's method that gives best results, and hence it is widely used, called as a Hucker and Korn modification in 1921. So it is a method that is we today adapt for gram staining. Here again, you have to explain the procedure, observation, mechanism of gram staining, and applications. Here in mechanism of gram staining, you have to explain four theories. There is lipid content theories, peptidoglycan content theory, magnesium ribonucleate theory, and Stern and Stern theory. So here in procedure, whenever we follow our general regular gram staining protocol, gram positive bacteria appears as a violet color and gram negative bacteria appears as a pink color or red color depending upon the type of counter stain that you use. Then second differential staining is the acid fast staining. You have to explain here the principle, procedure, mechanism, then its applications. This acid fast staining helps to differentiate acid fast bacteria from non-acid fast bacteria. Now see, this acid fast bacteria, they possess mycolic acid, which is nothing but a waxy substance into their cell wall. And these the examples of acid fast bacteria, they are mycobacteria. In that, Mycobacterium tuberculosis, Mycobacterium lepri, then several Nocardia species, they are acid fast bacteria, and they retain the primary stain. They never decolorize with the decolorizer, and hence they appear, they retain the primary stain, and hence they are called as a acid fast bacteria. Whereas non-acid fast bacteria, they are also stained by primary stain, but they are decolorized because they do not contain mycolic acid, and hence they are easily decolorized, and then they take counter stain and appears depending, and they appears uh, in color with respect to the nature of counter stain that is used. After principle, you have to explain the mechanism, role of primary stain, role of decolorizer, role of secondary or counter stain. So this is a procedure. Take a clean grease-free slide, prepare a smear, then flood the smear with carbon and you have to steam it. You have to keep it either on water bath for steaming purpose. Then decolorization is achieved by using acid alcohol mixture. And after decolorization, you have to add methylene blue, and then you have to air dry the slide, put a drop of cedarwood oil, and observe it under microscope. So acid fast bacteria, they appear as a red in color, whereas non-acid fast bacteria, they lose the primary stain and they take up the methylene blue as a counter stain, and they appear as a blue in color. So observations: bright red colored acid fast organism or blue colored non-acid fast bacteria, depending upon the type of organism that is present in the smear. Applications: they are used in differentiation of these two types of bacteria. Then they are used in detection or diagnosis of TB, leprosy, etc. Then special staining procedures, they are used to stain special cell structures, hence called as a special staining procedures. In our BSc1 syllabus, there are three examples of special staining. First is the staining of cell wall. And here you have to discuss principle of cell wall staining, then procedure of cell wall staining as a chances method, then observations, then mechanism of cell wall staining, and then applications. Here is a flow chart that explains the procedure of cell wall staining. I think there is no need to go in detail because we have taught these concepts many times. Then staining of metachromatic granules. These metachromatic granules, whenever they are stained with blue stain, they appear as a red in color, so they are called as a meta, that is change in color. They show change in color, so called as a metachromatic granules. They are also called as holutin granules because they were first discovered in Spirillum holitans. Then they are also called as a Babson granule. They are chemically made up of polyphosphates, hence called as polyphosphate granules. They are found in Corynebacterium diphtheria, Yersinia pestis, Mycobacterium species. You have to explain here the procedure which is mentioned here in the flow chart. Then here this flow chart is for volatility granule staining. Prepare a smear on slide, air dry and heat fix the smear. Then here one step is not mentioned here, that is removal of fat. Removal of fat with the help of solvent, because here we use the curd sample. We use the curd sample and hence it needs the fat removal. Then you have to treat the smear with Albert stain for 3 minutes. Then drain the excess stain and flood the slide with Albert's iodine for 2 minutes. Wash the slide with water, then air dry and observe the oil immersion heads. The observation is granules appear blue-black colored, whereas cell looks green in color. So this is a procedure and observation of metachromatic granule staining mechanism is mentioned here with applications. Albert stain helps to distinguish Corynebacterium diphtheria from most of the short non-pathogenic diphtheroids which lack granules. Then third special staining is the staining of cell wall. Staining of capsule. So there are two staining methods: positive staining and negative staining. Here, negative staining, that is, Manuel's method is used for capsule staining. Here, the procedure is mentioned as per this flowchart. Take a clean, grease-free slide, put a drop of Congo red color, mix a bacterial culture, prepare a smear, then air dry the smear, put a drop of Manuel's stains, then air dry and add oil immersion lens and observe under the oil immersion lens. And here the observations are: capsule appears colorless against blue background, whereas the bacterial cells appear red color. Here the mechanism is mentioned here with this method makes uses of two reagents, that is Congo red and manual stain. This Congo red, it stains background, whereas this manual stain, it contains 10% FeCl3. It acts as a mordant. 5% phenol increases the penetration of the stain through the capsule, and acid fuchsin, it is a stain that enters into a bacterial cell, or it is adsorbed onto the bacterial cell surface because of action of 5% phenol, and acetic acid decreases the pH to acidic side, and hence due to acidic conditions Congo red appears as a red. So the observations, they are as predicted. Then applications: capsule staining allows us to classify the bacteria on the basis of presence or absence of capsule, and this capsule detection, this capsule, it gives virulence factor to a cell, and hence in order to differentiate in between the capsulated and non-capsulated cells, there is need to detection of this capsule. Then last unit is the control of microorganisms. So this is unit 4, that is control of microorganisms. These are the points that are included under the syllabus. Definitions: use of several physical agents in control of microorganisms and also several chemical agents. Then learning outcomes: after the completion of this unit, student will be able to understand the concept of sterilization, disinfection and sanitization. Then study function, application and advantages of different physical and chemical agents. In this unit, you have to explain the definitions of some of the terms that are important in control of microorganisms. They are sterilization. It is a process of destroying all life forms. Here, vegetative cells and spore-forming microorganisms or spores. Both these two forms, that is vegetative cells and endospores, that are destroyed, and hence it is called as sterilization. Disinfection, it is a process of destroying only vegetative forms of pathogens and not the pathogens, and hence it is called a disinfection. It kills only the vegetative forms. Then antisepsis, it is a process that kills only growing cells but not the spores of pathogens. Now see the terms disinfection and antisepsis, they are similar. The difference is that these disinfectants, they are used for non-living surfaces, whereas antiseptics, they are used for living tissues. That means in order to kill the growing cells of a pathogen from non-living surfaces, disinfectants are used, and in case of living tissues or cells, antiseptics are used. Germicide, it is any substance that kills germs that cause disease, is called as a germicide. Then microbiostasis is a phenomenon that inhibits the growth or multiplication of microbiota, is called microbiostasis. Then sanitization, it refers to cleaning and disinfection of an area or an item, is called as a sanitization. So these are terms that are important in control of microorganisms. Now let's see the different physical agents that are used for control of microorganisms. First factor is the temperature. Here you have to explain the importance of temperature into the environment. Then this temperature, each organism that exists into the nature, it has minimum, optimum and maximum growth temperature requirements. And beyond the maximum range, the temperature have killing effect on the microorganisms, whereas the temperature below a minimum inhibits the microbial growth. So these are called as a cardinal temperature ranges, and these are for all different kinds of microorganisms. As compared to vegetative cells, we all know that endospores are much more resistant to heat than vegetative cells. Vegetative forms are killed within 5 to 10 minutes at 60 to 70 degrees Celsius, whereas endospores require temperature above 100 degrees Celsius for prolonged period. Please note that in presentation, the degree Celsius sign is mentioned. It is a subscript here. It should be superscript degree Celsius. Degree sign should be superscript. So you have to explain here the importance of temperature and the temperature requirements of microorganisms. Now see some terms that are used to evaluate the effectiveness of heat, because heat is used to control the microorganisms from the environment. So thermal death point, it is the lowest temperature that kills standard suspension of organism in 10 minutes. Then thermal death time, it is the shortest time that kills standard suspension of organism at a definite temperature. Then decimal reduction time, it is the time in minutes required to reduce the population by 90%. So these are the TDP, TDT, DRT. You have to explain while teaching. Then heat is the most widely used method of sterilization, and it is employed in the two forms, that is moist heat and dry heat. Usually the moist heat is used to control the microorganisms by boiling in water, fractional sterilization, steam under pressure and pasteurization. Whereas dry heat is used in the form of hot air oven and incineration. So first, the temperature as a moist heat. So you have to explain here the mode of action of moist heat, as it kills microorganisms by coagulation and denaturation of enzymes and cellular proteins, and it also degrades nucleic acids and it disrupts membrane. Then moist heat, it is employed in four ways. First is the boiling in water. Here you have to explain this method with respect to the process, then applications, then advantages and disadvantages. So you have to explain these points in relation to this method. Then fractional sterilization, which is also called as a Tyndallization, because it was described by scientist Tyndall in 1877. So this you have to explain here the definition of fractional sterilization, as it involves steaming the material for 20 minutes for 3 consecutive days with an incubation period of 24 hours at room temperature. So for day 1, you have to autoclave it for 20 minutes, then you have to keep it for room temperature, incubation at room temperature for 24 hours. Then next day, you have to again steam it for 20 minutes, then 24 hours room temperature incubation, then again sterilization. So this sterilization, it is carried out for 3 consecutive days. So it is called a fractional sterilization. And here, during 24 hours room temperature incubation, whatever the spores that are present, they germinate into the vegetative cells and they are killed in next cycle of sterilization, which is carried out at second day, third day. Then after the definition, this fractional sterilization, you have to describe this method, then you have to give its applications, then you have to mention the disadvantages, that explain the disadvantages as well. It is time consuming. This method will fail if the spores are present and they do not germinate during 24 hours room temperature incubation. So these are the disadvantages. Then third method is the steam under pressure. It is called the autoclaving. It was used in 1884. Chamberlain designed and used the autoclave for this purpose. You have to explain this method here that we follow regularly for the sterilization of our media. And we all know that steam, it has a great penetration power, and in autoclave, sterilization is achieved at 15 psi, that is 121 degrees Celsius for 15 to 30 minutes. In autoclaving, you have to take two precautions. Number one, you have to evacuate the air from autoclave. So whenever you place all the material for autoclave, you have to remove the vessel and you have to on the gas. So whenever whatever the air that is present in the autoclave, it will the boiling water that is present in the autoclave, it will start to boil. It will produce the steam, and the steam will release the air that is present in the autoclave. And once there is continuous flow of steam, then the you have to keep the uh, pressure regulating device or the vessel that we use for our regular pressure cooker. Because if air is present in the autoclave, then this air, it can this autoclave can raise the temperature, or it can attain the temperature in a pressure cooker only at 112 degrees Celsius. It will not go to 121 degrees Celsius. So you have to evacuate the air from autoclave. Then second precaution, the steam must have access to the material that is to be treated. So these are the two precautions that are to be taken while we operate the autoclave for sterilization purpose. Then you have to mention the applications that it is used for sterilization of material, anything that is not damaged by steam and high temperature. Solid and liquid media, discarded cultures, aprons, contaminated media can be sterilized by this autoclave. Then you have to explain the advantages and disadvantages for steam under pressure method. Then pasteurization. You have to define the pasteurization, and we all know that it is a process of heating every particle of milk at 62.8 degrees Celsius for 30 minutes or 71.7 degrees Celsius for 15. So as you increase the temperature, the time period is reduced. So this method, it was discovered by Louis Pasteur, and hence it is called as a pasteurization. It kills pathogens, spoilage causing microorganisms, and preserves flavor and nutritive values. And in methods, this pasteurization, which is usually applicable to the milk, it is carried out by low temperature holding, that is achieved at 62.8 degrees Celsius for 30 minutes. Then high temperature short time method, which is performed at 71 degrees Celsius for 15 seconds. And third method is the ultra high temperature, which is performed at 143 degrees Celsius for 1 to 3 seconds. So you have to describe these three methods. Then you have to give the applications, advantages and disadvantages of this method. So this is the fourth method, which employs the steam for control of microorganisms, moist heat. Then dry heat. This dry heat, you have to explain here the mode of action and methods. Here the mode of action, dry heat, it acts mostly by oxidizing or burning the cell materials. And here in dry heat, two methods are used. One is the incineration. These are the pictures of, uh, these are two different designs of incinerator that are used for burning the cell material. Here you have to explain the applications and disadvantages of this method as well. Then second is the hot air oven, which was proposed by Pasteur in 1876. The method uses the hot air oven. The temperature used is 140 degrees Celsius for 1.5 hours, or if you increase the temperature above 140, that is 180 degrees Celsius, the time is reduced from 1.5 hours to half an hour. So here you have to explain the working applications and disadvantages of hot air oven and how it is helpful in controlling the microorganisms. Then we studied the temperature as a first physical factor. Now see the second physical factor, that is a filtration. Here in filtration, the filters are used that removes the microorganisms either from liquid or gas. Then types of filters that are used for filtration. Here filtration, the microorganisms that are usually trapped over the filter, and the material that is filtered through these filters, the liquid material or gas material that is filtered through these materials, it traps the pathogens, it traps the microorganisms, and hence the filtered liquid or filtered gas, it is free of, or it is, we can say it is sterilized. So types of filters that are used, there are two types of filters. Number one is the depth filters. It consists of thick layer of fibrous or granular materials. And here the examples of depth filters are asbestos filter, membrane filter, porcelain filter, diatomaceous earth filter, sintered glass filters, or fluted glass filter. Here, among these filters, asbestos and membrane filter, they are in our syllabus, and you have to explain the working and applications of these filters. So in asbestos filter, the best known filter is SIS filter. Then second is the membrane filter, that is we generally use for water purification. These membrane filters are composed of cellulose nitrate, cellulose acetate, polycarbonate, and polyvinyl difluoride. These membrane filters, they are available in 0.01 to 10 micrometer range. 0.2 micrometer filters, they are most commonly used for sterilization. The membrane filter that is used for air sterilization in pharmaceutical industries and also in our laminar airflow cabinet are HEPA filters. And these HEPA filters, they have zero point 0.3 micron porosity, which gives 99.97% efficiency. Then these are the different types of membrane filters and filtration techniques. Then you have to explain the advantages and disadvantages for the use of membrane filter. Then next, third physical factor is the desiccation. Desiccation means drying. Desiccation means drying because we all know that the water is essential for metabolic activities of the microorganisms in their cells and also from their environment. So desiccation, whenever you remove the water either from, whenever you remove the water from microorganism or environment, the microorganisms dies. So here mode of action is dehydration of microbial cells and their environment, ceases metabolic activities, and finally kills microorganisms. So this is mode of action. Then the applications are, they are useful in preservation of grapes, figs, apricots, grains, leafy vegetables, and milk. The disadvantages of desiccation are does not sterilize materials. Some pathogens remain viable for longer periods. But generally, it is used for preservation of these grapes, figs, and we get the dry fruits and desiccated material. The next physical factor is the osmotic pressure. Here you have to define the osmotic pressure as a pressure that develops when semi-permeable membrane, that is cell membrane, separates two solutions of different concentrations. So if you take an example of a bacterial cell that exists in different environmental conditions. So the cell membrane is a semi-permeable membrane. The cytoplasm, it is its inner environment, and outside environment is different. So these two environments, whenever they differ in solute concentration, there is a flow of solvent from high solute concentration to low solute concentration. And during this transport, the pressure that exerts on a cell membrane is called as a osmotic pressure. So larger the differences in between the solute concentration in environment and a cell, larger will be the pressure, and hence larger will be the pressure on a bacterial cell membrane. So mode of action, plasmolysis and plasmoptosis. These are the two phenomena that occurs in two different environmental conditions. So this plasmolysis, it occurs in hypertonic environment. Hypertonic environments means salty conditions or high sugar concentration conditions. Here, the solvent solute concentration is high in the environment and it's very low in the cytoplasmic content. And here the water is a solvent. Water is a solvent. It will move from low solute concentration to high solute concentration, and it will leads to the shrinkage of a cell. And higher the difference in between the solute concentration in the environment and cell, higher will be the shrinkage, and finally the cell will die. And this plasmoptosis, it occurs in hypotonic environmental conditions where the environment have low solute concentration, and hence the water will move from environment to the cytoplasm, and hence cell will swell and it will burst whenever it goes beyond a limit. And this phenomenon is called as a plasmoptosis. And these mechanisms, they are taken into considerations while preservation or while control of these microorganisms. So based on this mechanism applications, they are useful in preservation of certain foods, jams, jellies, which are preserved by achieving high sugar concentration in the environment, whereas fish pickles, they are preserved by salty conditions in the environment. Then disadvantages, most of the microorganisms do not exhibit distortion by plasmolysis and plasmoptosis, and hence they will not be controlled or killed. Hence this is a disadvantage of this method. So here you have to define the osmotic pressure, then you have to explain different modes of action, plasmolysis, plasmoptosis, then applications and disadvantages. Then next physical factor is the radiations, UV rays, gamma rays. So electromagnetic radiations, they are of two types, ionizing and non-ionizing. These non-ionizing radiations, they possess moderate energy, so they do not ionize the molecules of, they do not ionize biomolecules, and hence are absorbed quite specifically by proteins and nucleic acids. The examples are UV light, which has a wavelength of 15 to 390 nanometer. And here you have to explain the mechanism of action of UV light, then its applications, advantages and disadvantages. So UV light as a non-ionizing radiation is to be explained to students with respect to these points: mechanism of action, applications, advantages and disadvantages. Then ionizing radiations, the example is gamma rays. And this process is called as a cold sterilization, because whenever you preserve or you expose the material with the gamma rays, it do not generate heat, and hence it is called as a cold sterilization. Then you have to explain here the applications, advantages and disadvantages of ionizing radiations. Then chemical agents for control of microorganisms. Different chemical agents are used to control, that is for sterilization and disinfection of microorganisms. Here, first one is the phenolic and phenolic compounds, which includes detols. Then you have B alcohol, then halogen compounds, chlorine and iodine. Then you have heavy metals. Then you have different gaseous agents as ethylene oxide, beta propyolactone, and formaldehyde. So all these 5 chemical agents are to be discussed while teaching with respect to their mode of action, then applications, then advantages and disadvantages of these chemical agents. These are the books recommended for this bacteriology paper. So thank you. Thank you once again, madam. Once again, I thanks organizing department and also the principal sir for giving me this opportunity to work as a resource person on this FDP for revised syllabus workshop. I am also a student of microbiology. We are all teachers. As I said, I have discussed my views on this paper. So thank you very much to you all.
Thank you, sir. Now I take an opportunity to express vote of thanks. So I would like to thank today's speaker, Honorable Dr. V.S. Patil sir, for conducting a very informative session and a valuable guidance on syllabus of the BSc 1 paper 2 bacteriology. So thank you so much, sir. Then
Thank you, madam.
I would like to express my gratitude towards organization or organizer of the FDP program, POS chairperson, Dr. S.S. Kanse ma'am, and head of the microbiology department, N.S. Joshi ma'am. I would like to thank all the faculty members for attending the session in offline mode. So once again, thank you so much all. Thank you, sir.
Thank you, madam.